Thanks for the nice introduction. And yeah, as just has been mentioned, I would like to talk a little bit about this new collaborative effort, also between actually three infrastructures: Infrafrontier, Euro-BioImaging, and the biobanking research infrastructure BBMRI. And this is about in vitro resources for preclinical research. I will start by first giving a little bit of background about Infrafrontier, and then talking more about the PrimTech 3R project. Let's see if I can move my slides. Yeah. I guess I preach here to the converted, but what we would like to what we like to show in the beginning is that we are talking about research infrastructure and research infrastructure projects. So likened, or we like to compare that to an airport. So we are not building the planes, but we are actually building the airport, and then different projects, smaller planes, larger planes can land on this airport. In our case, so it's Infrafrontier is the research infrastructure about European research infrastructure on modeling human diseases. So we provide resources, scientific resources, services, and expertise to advance our understanding of human diseases using disease models, mainly rodent models. We are a consortium of 21 partners in 15 countries, and I can show this here. And this is actually now the new map of the ERIC. Infrafrontier as a consortium, but also as a legal entity, is around since more than 20 years now, and there was an in-between legal entity, but now we have the ERIC, and you see here in dark blue the member countries. This would be Germany as the hosting country, France, Czech Republic, Greece, Sweden, and Finland. We have now Spain, beginning from next year. Spain will be an observer. And what you can also see here on the left side, and also depicted by these numbers, these are the national nodes. So the dark blue ones are the national nodes that are from the ERIC member countries. But then we have also these yellow countries, which is our, let's say, the bigger consortium. And these are also countries that provide active or are active service providers in Infrafrontier. And so actually our network is larger than just the ERIC, the current ERIC members. And of course, we hope to basically, now that we have the ERIC in place, enlarge it also to all these other of our countries. So this is the team of the headquarters, and I would today particularly highlight also here Azra Ali Khan. He's our scientific project manager. He was also very much involved in setting up the PrimTech 3R projects that I will present today. So Infrafrontier's core resources and services are around rodent models for studying human diseases. So, and in this respect, the model generation, so creating different genetic models, this is a major part. We have the systemic phenotyping, which are large scale analysis facilities where the functional genomics of different genetic models can be studied. The longer standing Infrafrontier resource is the European Mouse Mutant Archive, or EMMA, which is a biorepository, a genetic repository of genetically altered mouse strains. And currently we have more than 8,700 strains in this European mutant mouse archive. And then we have more specific services also for different disease areas. And we have exenic services. So these are mice with defined microbiota, which can be then used in microbiota research, and we have BSL-free labs, meaning very clean labs for also for studying infectious diseases. But we are also apart from having this let's say the core of the rodent models, we are also more and more venturing or flexing our muscles into the in vitro models, and this is something that I would like to talk more today. But before we come to the in vitro models, maybe still one step back and having a look at mouse models in biomedical research in general. So mice have been used as a model for human diseases for a very long time because of their high genetic similarity. So over 90% of human genes associated with diseases have also counterparts in mice, and many breakthrough discoveries on human diseases have been achieved in mice. And then there's a very advanced genetic toolkit also recently also with the CRISPR-Cas and other gene editing technologies that are available in mice and that we also make use of in Infrafrontier and offer to the community. And in biomedical research, I mean, mouse models are also, of course, what the most widely used animal model in biomedical research, and particularly also in preclinical research. Also because there's ethical and legal requirements, and in the drug testing and the preclinical testing pipeline, very often so larger animal models, but also small animal models like mice and rodents are mandatory steps. And so the majority of all laboratory animals used in preclinical research are rodents. So now venturing slowly into PrimTech 3R. So as I just said, preclinical studies are heavily reliant on mouse models, but industry are also looking and also asking us for cost-effective, reliable and scalable alternatives to the in vivo models. To use in specific preclinical testing pipelines. And so Infrafrontier has been, or Infrafrontier partners have also been approached by different pharmaceutical companies asking for specific models, specific also in vitro models that can replace the in vivo models in this preclinical testing, and this is basically the PrimTech 3R project is our response to that. So now about PrimTech 3R. It's a European Commission funded, so it's an EC funded project, and it aims to provide preclinical in vitro models for the replacement, reduction, and refinement of mouse models in preclinical studies. And so what should be the outcome, or let's say what's being developed in the context of these projects are so-called complex in vitro models, or CIVMs, that then can replace, reduce, or refine existing tests, preclinical tests in preclinical research. I will come to this complex in vitro models in a second. So since we have also the PrimTech 3R has the 3R concept explicitly also in the project title. So it's about replacing, reducing, refining. So replacing in vivo models in preclinical studies by validated and characterized complex in vitro models. Reducing the use of the in vivo models by providing comparable in vitro models that can be created via biobanked cells. and refining the process of obtaining and storing mouse cells from mice, leading to standardized and reliable procedures for their acquisition. As I already mentioned, this project is carried out together or in collaboration with BBMRI, Eric, so the biobanking research infrastructure, and Euro-BioImaging. And I guess one important aspect also to mention here, so we are mostly, I have to say, but we are talking about mouse-derived or rodent-derived in vitro models, because this is basic. So because what we would like to replace or refine or reduce is the use of mouse models, rodent models in preclinical research. Just very briefly on this what we call, or what are called complex in vitro models. So these are cutting edge in vitro systems that also achieve this higher level of cellular complexity and therefore also more closely mimic the situation in certain diseases. And so basically, so if we are talking about let's say basic 2D cellular models, so now the in vitro the CIVMs, the complex in vitro models have higher complexity and higher physiological relevance. And here are some examples. So transfer system organotypics, organoids, tissue explants, microfluidics. In the PrimTech 3R projects, we have a list of CIVMs or complex in vitro models that should be either refined or developed in the context of this project. So we are talking about lung butt microarrays, 3D co-culture glioblastoma models. Then also bioprinting is involved, 3D printed muscle fibers in tumor on chip. 3D bioprinted mouse colorectal cancer tissue, epidermis models, so human 3D organotypic epidermis models, GLUT1 deficiency syndrome, blood-brain barrier complex in vitro models, and then also air-liquid interface lung organoids. So this is the range of complex in vitro models that we're looking into in the PrimTech 3R. And the way these CIVMs are being then also developed and tested and also validated is shown here in two examples. So these would be our so-called demonstrators. So there are certain demonstrators. And here in this, for instance, here, this would be the colorectal cancer demonstrator, where the cells would be, so in one task, they are isolated from mouse cells. Then they will be tested in bio or drug testing will take place in bioprinted colorectal cancer tissue. And then also there is the in vivo validation where these CIVMs are then compared to mouse models from Infrafrontier. And the same is here an example for the epidermis models. So this is how we organize this let's say this developmental pipeline and validation pipeline in PrimTech 3R. And at the end of the project, what we hope to achieve is a shared in vitro resource, and shared with shared, I mean shared also between the three research infrastructures, so between BBMRI, Infrafrontier, and Euro-BioImaging. And this in vitro resource has several components. So there's the pilot mouse biobank. So this would be In addition to our genetic repository, this would be then a biobank of suitable mouse cells. Then we will also have standardized protocols. We have the validated complex in vitro models. Then also guidelines for comparing in vitro and in vivo models. Cellular disease models, and then also a lot of knowledge, staff training, and the online manual. And this together brings up the PrimTech 3R in vitro resource. Just very briefly also, so about the role of Euro-BioImaging in this collaboration. So it's the detailed characterization of the CIVMs for drug responses. Two partners will be specifically involved. This is Dr. Daniel Rabada from IPCAS, Czech Republic, and Professor Morten Nielsen from Aarhus University. And here you see also their roles in the project. And just as one of my last slide, or almost the last slide, so this we have as Infrafrontier, we have a long-standing collaboration with Euro-BioImaging. So you see here different projects, EC-funded projects in the context of data, harmonization of data, in the context also of harmonization of services, then more specific projects on infectious diseases and cancer. But we think this is a new area also in the collaboration, because in most of these projects we have been working together on the level of the, let's say, the research infrastructure hubs. And also these are usually multi-array projects. And now in this case, this is also, we are very happy also to venture into common technology development, creating this shared resource, and also working together more closely also with the Euro-BioImaging national nodes. And I guess with that, I'm at the end. So this is a part of the Infrafrontier crowd. So thanks a lot for your attention. And we are very much looking forward to this collaborative effort. And I guess the only thing that I forgot to mention, it's a bit shown here. The entire business will start in April next year. Okay, thanks a lot.